Wonderful. Thanks everyone for joining today.
Yeah,
I'm excited to speak with you all and talk about Gtsummary, one of my favorites, obviously. This is why we're here. And I'm joined by Shannon Pileggi, who is, Shannon, would you like to introduce yourself briefly?
sure. Yeah, I'm Associate Director of Data Science at the Prostate Cancer Clinical Trials Consortium.
All right, thanks, Shannon. So she'll be here helping us through everything, making everything go smoothly. And also Emily Zaber is here from our medicine, also ensuring everything runs smoothly. I'm Daniel Sjoberg, and I work at Genentech. It's a biotech company. And right now, my role, I've recently switched from like a data science innovation type role to a role that does like real-time analysis of our trial data as it comes in to make sure all of everything's running smoothly and safely and collecting all the information that we need. I mean, no one's getting harmed by any of our treatments, that kind of thing. So thank you for joining. So this, Shannon already dropped the URL into the chat, so that's great. I recommend you open up the website now. If you are going to be following along with some of the exercises, some pre-work that you can do is installing the tidyverse and Gtsummary. That should get you set up for success when it comes to the exercise later on in the presentation, in the workshop. Just to give you a little bit of navigating what you're, so this website, it will be up and running. It's live now. It will continue to be live forever as long as GitHub is freely hosting websites for us. Over here on the left, you can see there's the homepage. Then we have exercises and solutions, and then a license. And then scroll down a little bit, and you'll see here the slides, and then a little bit about Shannon and myself down at the bottom. So I think we can get started. So we're going to be talking today, obviously, about Gtsummary, but a little bit more in depth, like what if you want to be using Gtsummary at scale in a large organization or a small organization, but you want to be incredibly fastidious about the way you QC your tables, how you prepare your tables, and these kinds of things. So a little bit more of the infrastructure. We're going to start with some of the basics, but then move into a little bit more of the infrastructure on how you can incorporate some more processes into your workflows within your team to ensure reproducibility and a high quality of exactly everything that you're working on. So for example, I used to work at Sloan Kettering Cancer Center, where Gtsummary was originally born, and now I work at Genentech. So it's owned by Roche, a very, very large pharmaceutical company, and Gtsummary is actually being used for the reporting for every single trial at the company right now, or it's rolling out at some stage of rollout in that organization. So of course, in an organization like that, there's a big emphasis on making sure fidelity and accuracy and reproducibility. So I want to talk some about those things that we do there on that side. So just real quickly, this is all unlicensed under Creative Commons, so you're free to use these materials for anything you'd like in the future. Just acknowledge the source if you do. If you'd like to connect on any of these platforms, please do so. I will, I have been sharing these links for a while. If you message me on LinkedIn, I'm terrible at LinkedIn messages, so I apologize in advance. I will eventually get to the connection requests, however. And I'm not as active on Blue Sky as I used to be on Twitter years ago. So, but I would still love to connect. So just want to set some expectations there in the beginning. And then Shannon, do you want to say anything here as well?
There's not much to say. You can go to my website to find more information about me. I'm also on Blue Sky or LinkedIn.
Right. She sounds like she's a little bit more active than I am. So good job. We're a relatively tight-knit small group here, so please feel free to raise your hand, ask questions at any time. In the interim between questions, however, remaining muted is helpful just in case there's some background noise, so we don't get to hear you eating your lunch, for example, or whatever else is going on at home. All right. With that, please pop questions in the chat. With the layout right now, I don't have a good visibility at the chat. So Shannon, please, if there's a question that someone pops in, interrupt me, absolutely. So let's get started. So Gtsummary got started, in 2019, I believe is when we made our first release. And really, I wanted to address some reproducibility issues on my own team and at Sloan Kettering. We wanted to build something that was like very, very easy to use, but incredibly customizable as well. And that's really what was the beginning of the Gtsummary package. So some pretty humble beginnings, I would say. But how is it going now? It's gotten over 200 installations. Excuse me, that's 2 million, not 200. 2 million installations from CRAN, 1,200 GitHub stars, over 1,200 citations and peer-reviewed articles, 50 individual code contributors. Last year, it won the Brian Bowl Award for Excellence from the Pharma organization, or the Open Source and Pharma organization. I think they're in the middle of a rebrand. In 2021, it won the American Statistical Association's Innovation Award and also won an award from POSIT as well. So really, I think from those beginnings, it's become so much more than I ever thought it was ever going to become. Even apart from in this, medical space where I operate, it's become the most downloaded and most used package for summary tables across the entire R ecosystem outside of the medical space as well, which is pretty wild. It's been really fun, you know, working on a package like this that you get to interact with a lot of people across the world and across various industries as well. So just a quick summary of what Gtsummary does. So apologize in advance if you know the package well. We're going to start out with some of the basics to kind of give ourselves a common foundation to build on. But really what we're doing here is just trying to create summary tables that have very sensible defaults but are highly customizable. So really what the goal here is if you want to write 1 or 2 lines of code to get like a really nice summary table for your demographics, your table 1, cross tabulations, regression models, survival data, survey data, other kind of custom tables. You can do that very, very easily with very, very few lines of code, which is great. But of course, you need to make things highly customizable, and I think that we've designed it in a way that is incredibly easy to do and intuitive, as intuitive as we can make it, to make it also very powerful and customizable. So we'll talk a little bit about that. We're going to focus on creating summary tables, but there are some other things that you can do that we're not going to cover today, such as reporting things in line. So what I found my team and us doing quite frequently at Sloan Kettering was that you were making summary tables of these results from studies, what have you, and then we would write summaries of our reports in our Markdown or Quarto, and we were constantly referring to the tables to report the odds ratios, the hazard ratios, the proportions that we were reporting from those tables in the text. So there's something called inline reporting. And what that does are these Gtsummary functions that let you pull any number from the table and put it in line inside, open text of your Markdown reports or your Quarto reports. We're going to talk about how you can stack and merge any type of Gtsummary table and talk about themes later too. And of course, print engines, I think, is one of the best parts of the package. So for our workshop, we're primarily going to just focus on like 2 functions. TBL summary, it's like the powerhouse that can do a lot of different types of summary tables. And TBL hierarchical is going to be highly customized for hierarchy data. So that would include our adverse events are the most common one because you have like a system organ class that is on an umbrella for each adverse event term, for example. But also this applies to a lot of things. Drugs fall into classes. All sorts of procedures fall into various types of classes as well. So it's a more common data structure than you might think when you just think of adverse events, which is primarily what we wrote it for.
Daniel, if you can pause for a moment, there was something in the chat. If there's anything you can do to increase your mic volume, I'm going to guess no, that you're at max mic effect. But if you just want to check your settings real quick to make sure, I do feel like you're coming in fairly clearly for me.
There is an output volume. Is that louder?
I think it's the exact same.
Okay. Now it's at max output volume, but oh, that's for my speaker. Just a second. Let me put that back down before someone speaks and blows my ears out. My input volume for my mic is I think that might be everything. I apologize, but for the most part, are we doing okay?
Yeah, I can hear you okay.
Okay. All right. Apologies. Hopefully you can hear me okay. So while we're going to focus on these two functions, there are a number of other incredibly helpful functions for hierarchy tables, for cross tabulations, for continuous summaries, wider summaries, but we're not going to go into them today. So if you're interested, take a look. All right. So ahead of doing anything Gtsummary wise, let's prep some data. So I'm going to be using some data that's available from the Pharmaverse Atom dataset. So, I work in pharma now, so we're going to be using something called Atom datasets. Atom datasets are a standard in the pharmaceutical space for FDA submissions, but they're pretty universal. So we're going to be using one called ADSL. And what ADSL is it's a subject level data set. So it's going to be one line per subject. And we're going to be using ADAE. That's our adverse event. So that's going to be one line per subject per adverse event. And we're going to be using ADLB just a little bit near the end. That's our labs data set. So that's going to be one line per subject, per visit, per lab. And I think we have just one example with this oncology data set, and that is going to be one line per subject as well. So you can take a look at the details here, but yeah, all of this data is available from the Pharmaverse Atom package. So let's start with the simplicity. So this is what I really like about this package. You just load the library. We're passing our one line per subject data set to TBL summary and just saying, can I get a summary for these three variables? That is that simple. And in the background, there's quite a bit of work happening. So here you're getting your beautiful table. Like I could publish this, that's fine. Go for it, right? But what is actually happening in the background is that Gtsummary is taking a look at the age variable and it's saying, okay, well, it's a numeric variable. There's quite a bit of spread here. There's a lot of unique values. I think that's going to be a continuous variable. So I'm just going to display that on a single row and give you the median and the quartiles. Ethnicity, I believe it's a factor in this data set. So it's, of course, going to default to something like a categorical summary. And then female is a true-false variable. And that is going to be defaulting to a dichotomous variable. So dichotomous is very similar to a categorical variable, except for it just displays on one row where there are two levels. And so you show the level of interest, and then the other level is pretty simple to intuit in most cases. So that's what we're dealing with. So we've talked about continuous summary types, we talked about categorical summary types, and continuous summary types, right? There is a fourth summary type called continuous 2. And what continuous 2 is going to give you continuous summary statistics that are shown on 2 or more rows. And we'll get to an example of that as well. You'll see here that the default stats are median IQR and count and proportion for the categorical and dichotomous. You can also see that age is the column label for the variable, all capital age, and ethnic is the variable name, and ethnicity is the label. So Gtsummary not expects, but it will look much nicer if you label your data. And I always encourage people to label their data. It's so important for you reusing your data later. So that will be used if you have those. If you don't have any labels, it's just the variable name will appear here. All right. So let's start customizing this output, just kind of get an idea of how we do that. So we're working basically off of what we did before. We have our ADSL data set that we're passing in. We're including the same three variables, but this time we've added the variable arm. But quick question, when I highlight like this, are you seeing the same highlighting? Like I have by equals arm highlighted on my screen.
It's highlighted blue.
Okay, thank you. That's good. Always good to know. You never know what mousing things are going to appear on the screen share, which ones won't. So you can see here that rather than a single overall column for our summary stats, it's now by arm. So we have our placebo, our high dose, and our low dose active treatments there for you. So we've done that. So because that's a very common thing, of course. And I mentioned our type continuous 2. So rather than using the default where age is going to give us a median IQR on a single row, you now see that age is on 2 rows. And we're going to further customize that a little bit. So in this very basic customization, age is a header row, and below that, you have your median IQR. But now let's change those statistics. So for age, rather than just giving me median IQR, let's pass a vector and say I want my mean and standard deviation on one row. And then in the next element of that vector, you can see min and max. And so those are the two rows you're now seeing in the table. And for female, rather than just giving me the n in the count, can you give me the n divided by the denominator and then the proportion as well? So that's how you can customize. And we'll talk a little bit more about this syntax too in the next slide. And if you need to change the label, you can of course change the labels too. So rather than the default label of just age, this is age comma years, to just be very precise that we're talking about years of humans here. And of course, you can change the default precision that things are shown to. So what Gtsummary is doing in the background, when you just pass it the variable age, it's looking at the spread and just making a guess, like, okay, the variability looks like from here to here. Probably we can just round everything to the nearest integer. Or if you have a pretty tight spread, then maybe it's going to show you to 1 or 2 decimal places depending on that spread. In this case, it defaulted to give us the nearest integer. But here we want the standard deviation to be to 1 decimal place. So that's why we gave this standard deviation. Now this matches the function name that we're passing up here in the statistic. So it's saying SD equals 1. So you may give me 1 decimal place for standard deviations. All right. So that's kind of the basic syntax that you'll see in Gtsummary. A little bit more on that statistic argument. So if you've been working in the tidyverse, you probably recognize this curly bracket syntax that appears every so often. It's called glue syntax. And this glue syntax is actually doing double duty. So not only is it telling me how to put the mean and the standard deviation together, because right here it says mean all by itself, then standard deviation is going to be within the parenthesis. And that's exactly what you see in our resulting table as well, right? So it's also giving you the function name. So what it's going to do first is it parses this string and says, okay, I need to go find the mean function and I need to go find the SD function. And then I am going to go execute those functions on the vector age. I'm going to come back with those results. I'm going to format it the way that you specified in the digits argument or by the default ways. And then in the final step, it's going to do those classic glue insertion things where I have my calculated and formatted mean and I'm going to insert it right here. And I have my calculated and my formatted standard deviation and I'm going to insert it right here. So just that means that you can use all of the base R functions, any function from any package, or even user-defined functions as well, as long as they're available in your global environment or in the parent environment from wherever you're calling this.
So Daniel, I just want to let you know if I'm looking busy over here because there's a lot of great questions coming in the chat, but I don't think there's anything you need to address at this moment. People are getting excited about all of the options that are in the table.
Okay, great. I love options, right? Yeah, you can see a lot going on there. Sometimes I catch it, sometimes I don't, but yeah, just like I said, feel free to stop me at any moment. Okay, so in the previous slide, you saw a couple of ways that we are using this kind of, I feel like this kind of syntax was the first time it was ever used in the R ecosystem, but I'm not sure. But I know that GT has picked up some of this syntax as well, where you have this formula. And so everything on the left-hand side of your formula, that's tidy select. So you can put in bare column names like age, like in this first example. You can put a vector of bare column names, like in the type argument as well. You can use tidy select functions like starts with, any of, contains, matches, anything like that. And then also, we're going to talk a little bit about some Gtsummary specific selectors like all continuous. Now, what that lets you do is in the previous slide, I said, oh, for age, can you give me mean and standard deviation and min and max? But if you want to do that for all your continuous variables, you would use a selector like all continuous to say, do this for all of my continuous variables. We also have selectors for all categorical variables, all dichotomous variables, what have you. So the variable selecting happens on the left side. And then on the right-hand side of this formula is where you give the instructions. Say, okay, for age, I want the label to be patient age. For columns age and marker, I want the type to be continuous, and so on and so forth. So, and whenever you have 2 or more sets of instructions to pass, you just pop it into a list, right? So we also accept named lists. So this is really helpful in a lot of cases. But when you're talking with named lists, or you're utilizing named lists, you can't use tidy select. So oftentimes you'll need to use those selectors like particularly like all continuous and all categorical. then you cannot use that with a named list. But you can also mix them as well. So you could say, give me a list, all continuous, make my summary statistics mean and standard deviation, and then you could put in the second element of the list, age equals median and IQR. And so you've actually given instructions, two instructions for the same variable because age is continuous. So you said make them all median standard deviation, but then you've also followed up with like make age what is it called? What did I just say? Median IQR. So whatever occurs second, whatever the most recent instruction you gave is the one that's going to be shown in your table.
There are a couple of specific questions now.
Yes.
So when writing a custom function, does it need to know how to deal with digits, or does Gtsummary just accept my function's numeric output and format it separately?
Yeah, so all it does is it needs, so for this basic use, what you need is a function that accepts a single vector and returns a single number. So like the mean function, for example, brings in the mean vector, excuse me, the vector of age, and then it returns the mean age. So that's like the general format for any function that's going to be used in this more simple setting. There are more complex things we'll talk about in the future too.
And then there was a follow-up question. Just wondering, would it be possible to use an F string like mean colon 1F to control the statistics and formatting in one place?
An F string?
I know I wasn't as familiar with it either.
Oh, and let me see if I read the question and see if it gives me a little bit more context, but I don't think I follow. So
Okay.
just wondering, would it be possible to use an F string like mean colon dot 1F to control the statistics and formatting in one place? Oh, no. They're in two
Okay.
different places. Good questions. Thank you. I love questions, so please keep them coming. So we did talk a little bit about these all continuous and all categorical selectors. They're super helpful to be used within Gtsummary, not outside of Gtsummary. There's also another function called all stat calls. So you saw in our summary table where you had summary statistics for placebo and high dose and low dose. Those are all stat calls. So sometimes you need to do something that applies to those three columns only and not the labeling column that appears on the far left. So this is a nice way to do that kind of selecting as well. And in general, the Gtsummary function is going to work with a TBL prefixed function is going to create your initial table. Then we have add functions. So add functions are the second type of function that you're going to be using pretty frequently. And those are going to be adding additional columns of statistics or information. Sometimes they add a row, but most often there are times they're adding columns. Then you have your modify family. So modify family, you just pipe whatever came out of TBL summary straight into like a modify. That will help you change your table's headers, the spanning headers, the footnotes, bolding, italicizing, and more. And then we also have some very bespoke bold and italicize for styling, but you could also more generally control them from the modify, bolding, and italicizing functions as well. All right, let's give some examples of some common add functions you might be using. So I think one of the most common ones I use is add overall. So you can see here that I've done on lines 1 through 5 exactly what you've seen before. And I'm splitting it by treatment. And I'm adding on an overall column to just show what's going on for all subjects at once. That's a very common one. It also is quite common to add P. So in this case, I've missed my highlighting a little bit. So line 7, 8, and 9, I'm adding three different types of statistics. In add N, I'm adding that N column after the characteristic column. Add P is giving me a comparison between my placebo and active treatment arms. And then in add Q, I am doing a correction using the false discovery rate method. So, and those are all coming straight from p-adjust. So there's a lot you can do for some pretty basic statistics here. Now let's start doing some further customizations. So now we are doing TBL summary. I don't want to see the missing information this time. And in lines 6, 7, 8, and 9, oops, I am changing the titles, the headers from placebo and active treatment. I apologize. I'm doing my best to never pronounce this word in a public setting, and it's I don't want to, so I'm going to just call it the active treatment. So if you're getting confused why I'm saying I'm changing the header from active treatment, It's actually from this word that today's not the day to pronounce it for the first time, I don't think. I don't know. If someone knows exactly how to pronounce it, yeah, message me on the side with something very phonetic, please. So I'm using modify headers. So this was the modify family, again, are those functions that are going to modify headers, footnotes, spanning headers, a couple of other things. And I'm saying for column stat1, please make it group A, and for column stat2, please make it group B. Now you can see here that I'm using markdown syntax, and that markdown syntax for bolding is going to translate to our table as well. I'm moving on to line 10 with our modify spanning header. And I'm saying for all stat calls, now we talked a little bit about this function. This is a way to select all of the summary statistic columns at once. For all of my summary stat calls, can you make the spanning header drug? And again, using the double star for bolding here. And I'm adding a footnote as here as well. And I'm going to add it to both of my summary stat columns. And it's going to say median IQR for continuous variables and percent for categorical. So kind of just changing what the default was. So yeah, my highlighting got all messed up here. Apologies for that. So if you're wondering, well, how did you know that stat 1 was referring to group A and stat 2 was referring to group B, or placebo and active treatment? It's because there's a function called show header names. And if you print, it will print all of the column headers along with the underlying column name in the data frame. At the end of the day, we're just printing a data frame in a cute way here. So it makes it easy to use. So here's what that actually looks like. If you print this in your actual console, it prints out like with colors and it prints out a little bit nicer. So you can see here the column names for the 3 columns that we saw in our previous table were label with a header characteristic, with stat 1, which is now group A, and stat 2, which is now group B. Now there's a really cute thing you can do in here. These level n and little n, these are things that you can inject right into your header very, very seamlessly. I think the next slide shows us how. It does not. So let me quickly tell you what that means. So review the modify header function for details, the help file. But essentially what this does is you can put level in curly brackets and it will actually inject what the proper level is. And you can put in big N in curly brackets and it will inject this 168 and little n would inject this 84. So it's a really nice way to be able to utilize the information that's actually sitting back there in the background very, very quickly. So you're not hard coding things like if you wanted to do something as simple as remove the bolding from the header. So you would do modify header and say all the stat calls, just make it level in curly brackets without the double stars on the outside. And then it would just inject the levels right there. All right. Another wonderful add difference function is, oh, excuse me, an add function is the add difference function. So super common that you need to just, you're looking at 2 different types of treatment, 2 different groups, what have you, and you need to see the difference between them. So in this case, I'm just showing you between placebo and active treatment that the mean difference here is 7.6, the confidence interval is here, and the p-value for comparing that is here. You can do it for rates as well, whereas here you have 6.3, 5.1, difference of 1.2, and here's the confidence interval for that rate and the p-value for the comparison. And it also tells you here down the footnote what exactly the tests were doing so that it's very, very clear what's happening. Now you may want to change these because that's not the most beautiful label for or footnote for a publication, but at least the initial output that comes shows you something that you know exactly what you're looking at. So that's always important. And you'll see here that we now have this abbreviation that comes up. So all abbreviations that appear in your table are going to now have an associated, they're called source notes because they don't actually have a reference back into your table. So you'll see it doesn't have the 1 or the 2 in the footnote as a prefix. So these are all, they will collate them all and put them in alphabetical order. And so this one, you can see the 95% CI. You're like, oh my goodness, what is a CI? CI, is that a confidence interval or a credible interval? And you'll see in this case, it's a confidence interval. So easy peasy. Always trying to make the results very clear to you, no matter what you're doing. There's also a very generic function called addstat for just adding, whatever you need to do. So we won't go into any examples with it, but it's there in case you need it. So This is one of the things that I think can really level up your work using Gtsummary. And they're called ARDs. They're called Analysis Results Datasets. And they are very structured ways of saving all of your results, all of the numbers that you're seeing in a table in a, like I said, a very structured way for use in various places. So ARDs are now the backbone for every calculation in Gtsummary. So that means every time you see a number in Gtsummary table, there is a corresponding, very structured data set in the background that has that result saved in it, right? So that's really, really nice. And they can be extracted collectively or individually. So here I'm building your basic summary table from our ADSL dataset by treatment arm, and I'm including just one variable here, age. So you can see here that these things are called cards. So these ARDs, they're all created by a package called cards that I also maintain. So if you look into your summary, your Gtsummary table, and look at the cards object, you can see here that there are two, oh, sorry, let me also show you. TBL summary, then piped into add-p. So there's two things going on here. There's going to be two sets of ARDs, one for your base TBL summary table, and then another one for the add-p, for those p values. So the gather ARD function is going to, you give it your table object that you've saved, and then you're going to get a named list out of that, of all of the ARDs. So what you're getting out is your first named list, which is TBL summary, because that was created with the TBL summary function. And then the value of that list, or the element that's associated with that list, that part of the list is going to be the ARD that has all of the numbers for the summary table. And then you can see, and then the second element would be add p. And so here, I'm just printing what exactly an ARD looks like, just so that you know. So I am just plucking out the add p piece and then the ARD for age. So we were presenting the Kruskal-Wallis test for this particular, and we were only presenting the p-value, but the ARD actually has everything else you may need to know about it. So it has the test statistic, it has the degrees of freedom, and it has a text description of what that test is. Some other really lovely things about ARDs is that we'll talk about it later. Never mind. We'll get to it. We'll get to it. I don't want to get ahead of myself. All right. Any questions about ARDs before we move on? They're really a powerful thing for QC and the fidelity of your studies. All right. We'll talk a little bit more about them in the future too. So there's a lot more add-on functions that we're not covering today. So please go check out the documentation. The reference page just lists all of the functions that are exported from Gtsummary. There's also some really great articles here as well, talking just with lots of examples, all within the Gtsummary's website. So check it out. So that brings us to our first exercise. So if you go back to the website, and on the left-hand side, if you click the exercises button or link, and then there will be an exercise there, and it's going to walk us through creating a table with using the methods that we have just been using today. All right. So please get started and please ask questions. We'll give you 10 minutes. So this is the website, danieldsoberg.com/r-med-2016, 2026, my goodness, -GTSummary. You'll go over here to the exercises. See how it also has exercises and solutions. So, depending how adventurous you're feeling, please recommend doing this. But if you don't feel like doing it, you can also review the answers as well. So what we're going to be doing here is loading our data sets or our packages, and we're going to be using the pharma versus ADSL dataset to do a little pre-calculating of our data. And then we're going to be using TBL summary to make a summary table, and then changing the statistics that were presented, and then doing a few other things. So Shannon has reminded me that during exercises, although my default is to keep talking. I should just stay quiet. So thank you, Shannon. Not today, she didn't remind me. Years ago. Here I am. Still can't stop talking. All right, here it goes.
Daniel,
Yeah.
just gonna flag that to you. Okay.
There was a question in the chat. Can the superscript numbers of the footers be changed to other symbols? I think the function is GT's tab foot or tab footnote marks or something like that. It's in there somewhere where you can change them. And when we get to the print engine section, I'm going to give an example using the function as GT. Now, if I forget to mention footnotes at that time, somebody stop me and say, how does this apply to footnotes? And I will answer you. All right, we've got about a minute left. So there's a question about how to use superscripts in labels shown in tables. So there's a function called modify footnote, where you modify footnote body, excuse me, where you can add a footnote to any place in the table's body. And so you can add it there. You just have to like hone in on which column and which row you want, and then specify what that label is. Thanks for the question. All right, that puts us at time. So let's quickly go over what we're meant to be doing here. So we did a bunch of pre-processing of our data, got us ready to create our summary table. Use TBL summary to summarize age, age group 1, sex, race, ethnic, BMI, height, weight by treatment. So taking first the treatment variable TRT01A, that would go down here in the by, this is probably a little small, in the by argument. All of these variables here that we want to summarize, they would go down here in the include argument. So that would get us done with step 1. For continuous variables, present the following statistics: the mean, the standard deviation, and the median IQR, and the min and the max. So we've already been provided here the vector that we need to pass. So here in type, we will want to say all continuous formula tilde, and on the right-hand side of that, that mean vector. Oh, excuse me, for statistics. And then you want to also specify type continuous 2 for those variables as well, all continuous. And we'll look at the example a little bit later. Ensure that age group levels are reported to in the correct order. So I think you're going to need to make a factor to change the order of those before you pass it to Gtsummary. So you can see here a little hint here. Age group, make it a factor. And then the last thing, view the ARD saved in the Gtsummary table using gather ARD. So quickly down here at the bottom, you can see here that we put, before passing to Gtsummary, we specify the ordering of our age groups to make them print in the proper order. We want our table by treatment. We want to include those variables we discussed already. For all of my continuous variables, I want them to be continuous 2 to present on 2 or more rows. For the statistic, I want all of those continuous variables to present these stats instead of the default, which is just median IQR. Here, I changed the age group labeling. And I have down here, there's a bonus about putting active treatment above all of the columns for active treatment. So I had to use show header names to see what the underlying names were for those columns. And those were columns called stat2 and stat3. And so I'm saying modify spanning header, including these 2 columns. I would like there to be a header of active treatment. And then here at the end, I am gathering ARD to print the ARDs for these columns. All right. Any questions on that? Any discussion? All right. Let me move some things around and go back into full screen mode. One moment. All right. Thanks for sticking around. Next, I want to talk a little bit about the hierarchy tables, for adverse events, like we mentioned before. So we're going to be using TBL hierarchical for reporting of AEs, conmeds, and other types of things. Concomitant medications is conmeds. So the basic syntax here is that you're going to pass your ADAE dataset to the TBL hierarchical function. You're going to say, please give me those statistics by the treatment arm. And then the variables here, this is very important. This is the system organ class and the AE term that we are using. So that's the hierarchy. You're defining the hierarchy here. So the first one is the system organ class, which is a larger grouping than the AE terms. And you're telling us what is your ID variable here? What's your subject ID variable here? So that's going to help us calculate the proportions accurately. And then here you're telling us what data set I'm actually using for the denominator, because there's oftentimes, if you're working in some studies, not every subject is going to have an adverse event, right? So we won't actually know what the proper denominator is by just using the adverse event data set. That's why you have to give us something like the ADSL, the subject level data set. So you can see here, we did get our results by treatment up here. You can see here, this outer term here is cardiac disorders. That's the system organ class. And you can see we have rates here. You can see that within cardiac disorders, there are various types of adverse events you can report. Very, very easy peasy. And so it's all here for you. It's all great, all organized. So we just want to make these as a very, very basic adverse event table. It's presented all the time in publications and other reporting. So we just want to make that super simple. All right, ARDs and QC. So we're jumping around a little bit. So we're back to ARDs. ARDs are so, so wonderful for QCing Gtsummary tables. They are really, really wonderful because they also include the unformatted and the formatted numbers, right? So for example, for the mean of age, we're going to have 76.45321, but we're also just going to have the character 76, the formatted version. So like I said, you can extract the ARD from any Gtsummary table. And there are various ways you can QC things. But what you want at the end of the day is you want things to be easily QC'd. So in many, many situations, what you're doing is you're QCing a table at one point in your study, but you're often accruing more information all the time. So you want to set up your QC to be programmatic so that not only do you know that your data was accurate or on this day that you originally created your data set, but you also want to ensure that it remains accurate into the future as new data comes. And oftentimes what happens is new data comes, it's weird data, you didn't expect that data to come like that, and that's why having programmatic QC in place is incredibly helpful. So here we are, and one way to QC is to build your ARD, your Gtsummary table, extract the ARD, and rather than doing a programmatic reprogramming of the table and trying to do some comparison of the tables after they've already been built, what you can do is extract the ARD from your Gtsummary table and then build an ARD and then just compare the ARDs. Because those ARDs are just data frames, it's very, very simple to compare them. So let's take that adverse event table and let's gather the ARD from it. So you can see that this is 36 rows and 15 columns. And you can see that the grouping levels are here, group 1 and group 1 levels. So our grouping variable was the arm or the treatment arm. And we can see that we have levels placebo, and this is high dose, and this is low dose active treatment. And you can see that this is all for the system organ class of cardiac disorders. And then within cardiac disorders, we had our atrial fibrillation, I think it was, and you have your little n and your big N and your proportion. So you can see here that not only do we have the raw statistic, but you have the formatted version as well. So for example, the raw percentage is 0.012, but the formatted thing that you see in the table is scaled by 100 to be between 0 and 100 instead of between 0 and 1. And then it's been rounded, so that's just 1.2, right? So that's what you're looking at here. So it has all of the results that appear in that beautiful adverse events table that we showed right here, all of the numbers here, but it's in a setting where it's one row per statistic, and it's very easy to identify exactly which statistic you need. And as you can see here, we are presenting more data in this ARD than actually appear in the table because we are showing you what the denominators are in addition to the counts and the proportions. So how would I QC this table? So what I would do is I would go to the cards package, which we're really not going to talk a lot about today. We're going to go back to the cards package, and I'm going to use this stacking hierarchy function to build ARDs for the system organ class, but then also for AE terms nested within system organ class. You need both of those things. So that's why you have the stack name, because it's actually iterating over the variables in this vector to build me an ARD just for system organ class, then build me another one that accounts for all of this nesting. And then I, of course, need to pass it my subject ID so I can get my denominators correct, just like in the original table, and then pass what those denominators are. So there are various ways for comparing two data sets in R. So in the cards package, in the dev version that's available on GitHub, there is a function called compare ARD, and it really helps you just compare ARDs. It's, you know, it's not a complex comparison because it is just two data sets, but at the end of the day, that function has a lot of built-in default for that make it very easy to compare two of these things. So you can set up that comparison by building your Gtsummary table, building the ARDs separately, comparing those ARDs at the end, and just moving on from there. And that'll be really, really nice to programmatically forever have those checks in place. And you can set it up so that it can error if anything ever becomes out of sync between those two things. All right, are there any questions about this and ARDs and anything else? The reason I ask is that I see the chat doing this, but I can't actually read the chat. So if there
people are really excited about this function that they're learning about for the first time.
Ah, okay, great. It's cute. It's very helpful. So one of the things that I really love about Gtsummary tables is that you can construct very complex tables by first deconstructing them and creating their really basic parts. So it's easy to make easy tables and hard to make complex tables. But what we've done in Gtsummary is we have merging and stacking capability that make it easy to construct more complex tables by just building easy parts of complex tables and then putting it all together. So let's go through a couple of examples of that. So TBL merge, I think is pretty obvious. What it does is it merges two tables like this, left to right. So this is a very basic example, and it doesn't really show the full capability, I don't think, but I needed to keep the example in the functions that we were actually using today. So TBLN is essentially just the first two columns of this table. And TBL age, it looks like, oh yeah, is using the TBL continuous function to give us the age summary statistics by ethnicity and by treatment. So you kind of have this like not 2 by 2, but like N by M table here, and you're giving me median age and IQR of the age within each of those combinations. And so that's what that gives you. So it gives you that characteristic column and then also these later 3 columns about the dosing and or the treatment that subjects received. So what you can do is you can plop both of those tables into a list and pass that to TBL merge and tell it, oh, in this case, I don't want any spanning headers because the default spanning headers are really ugly. You don't want to see them anyway, but it also makes it very clear. So I usually remove them after I confirm that the results are exactly what I'm looking for. And you get this beautifully combined table. So if you ever needed to construct a table like this, again, I'm making 2 very, very simple tables and making a somewhat more complicated one by putting them together. So easy example. We will also move into a more complex example in just a moment. So stack is similar, except for that you're just going to be stacking tables instead of merging them horizontally, right? So in this case, I am making a table just for drug A, which is my high dose active treatment. And then I'm making another table that's just for low dose, drug B. So I have those 2 very, very simple tables, and I'm plopping them into a list, and I'm stacking them together, and I'm giving them a group header in this case, drug A and drug B. So you can see that this first part of the table is drug A, and that group header is being added right there. And then you can see that drug B appears just right below it. So again, very simple example, but I will show the power of it in just a moment. Oh, there's one more function I want to talk about, TBL strata for stratified tables. So oftentimes what you need to do is you just need to build the same simple table by, for example, treatment arm or by some other kind of stratifying variable. And this is a basic utility function. So let me just walk through this one real quick. So I'm passing it the ADSL dataset. I say, can you build me a bunch of tables by arm? And then here is where I'm actually building each one of those tables. So this is that formula syntax that you see all over tidyverse for anonymous functions. You can also just create a proper function here. And .x is representing the stratified piece of ADSL within each of the arms. So within each stratum. So this is a stratified or a subsetted table of ADSL. and I'm making a wide summary. So what essentially wide summary does is it makes you a table that gives you the characteristic column and then the N and the percent. All right, so for ethnicity, it's giving us the N and the percent within placebo only. And by default, it's going to just merge all those rules together. So what it did is it independently built this table for placebo, independently for high dose, and independently for low dose. And then it took those three independent tables and merged them together. So one thing that kind of trips people up about TBL Strata is that sometimes people think it's going to do a lot more than it's doing. This is a basic utility that helps you build three tables quickly and then merge them. It does not share context among those three tables. So sometimes if, for example, if in the low dose group, it was all Hispanic or Latino, and it wasn't a factor variable, then this individual construction of the low dose table wouldn't have a row for not Hispanic or Latino because there'd be no context for it to know within that subset of the ADSL dataset that there is a level for not Hispanic and Latino because all it knows is that they're 100% Hispanic and Latino in that subset, right? So it's important to keep that in mind when you are merging those things together. It's meant for merging like tables. They need to look similar. They need to have similar structures to get the exact structure that you're expecting. So in that case that I'm outlining here, if in low dose we only had Hispanic and Latino subjects, then what you would need to do is before you pass all of this, the data set to TBL Strata, you would have to tell ADSL, ethnicity is a factor and it has these two levels. Because if it's a factor, even if there are no observed levels in the low dose group for not Hispanic or Latino, it will still give you those zero counts. Right? So just when you're building with TBL Strata, keep that in mind. This is in the documentation, but I still get a number of questions about this. Actually, I'll take that back. Since AI has become so popular, I get so few questions asked to me, and it's just beautiful because AI does a great job of answering the questions. There's a question of how are missing handled, considering the three tables created by TBL Strata requires three similar tables. So by default, missing the missing counts are shown in the table if they are present. So if there are missing ethnicities within placebo but not within high dose, you would have two different structures that are returned to you. So you would need to add to your call to TBL summary, for example, exactly how you want missing values handed. So we do our own default thing in the background, like, okay, well, let's just see what happens in the background. And then we're going to push them all together. But if you know that there are going to be differential missing patterns among what you're reporting, then you need to tell me with the missing argument in TBL summary, for example, that, oh, can you always show me the missing row even if there are zero missing, or can you never show me the missing row even if there are missing levels of that variable? So while we do whatever we do in the background to kind of show you what's in the data, when you start merging things, they are meant to merge like things. So you need to add those additional arguments to make sure that you're merging like things. That's a great question. So by default, we're going to merge everything, but you can use this argument here to stack things as well. So this is the exact same example, but stacking instead of merging.
We have
right,
a
any questions?
similar question, but in the table regression context.
How do you address that issue if one factor level when merging tables you get from TBL regression where you don't have much control? Oh yeah, so if you have a regression model, so there's a TBL regression function that takes in many different types of regression models, and it will build tables for you. But what happens in regression models is if there is a level that's unobserved, the regression model will often just like cut out the level because like what is it doing there anyway? What is a regression model supposed to do with an unobserved level, right? And then you wanted to, so let's say have a regression model where you have ethnicity as one of the covariates. Ethnicity is going to be just like dropped from the model if you only have one observed level, right? But you have your TBL summary where you're showing the counts of each one of them, and maybe you want to show that zero level, that zero count. So in that case, you would have to do a little bit of munging on the inside to make sure that you get exactly what you want, right? So you're going to have a regression table that's lacking ethnicity. So when you merge them, ethnicity is going to probably appear at the bottom, where it has like the summary statistics from TBL summary, like the counts or whatever have you. And then when you look at the merged piece from the regression table, there's going to be nothing there, right? So you'll just have to do a little bit of munging. And like I said, we'll talk a little bit about how you can do that, but just very quickly to answer your question, a GT summary table is just two things essentially. It's a list. If you look at the internals, it's a list. One thing is a data frame, and it's just a raw data frame that we're going to print. And the next thing is a list of instructions on how to print it in a cute way. So that data frame is something you can modify directly oftentimes. So how, in various cases, if you need to add in a missing level that wasn't there, you can just go right into the level, right into that data frame and use a function from Tibble like add row and just say like right here, can you add a row if it's not already there? And you can just add it and then merge it. There are various ways to handle it. After you merge it, if something gets out of order because there was a mismatch, you can just resort the table to do whatever you needed it to do in the beginning. So there's a couple of ways to handle that when you are merging unlike tables. Just remember at the end of the day, it's just a data frame, so don't be scared. Even though we print it like a beautiful GT table, we're just printing a data frame, and you can modify that data frame in any way before it gets printed. So one time long ago in the beginnings of Gtsummary, I got an email from a Swedish group of researchers that's like, hey, loving the package, thanks so much. Can you add a table that looks like this? If you took Epi 101, you may be having PTSD right now, or, this is a very, very common table from Epi 101 and from grad school, not 101, I guess, like from grad school 101, whatever that means. So here we're looking at odds ratios and Cochrane-Mantel-Hansel adjusted odds ratios and p-values and counts for cases and controls, whether or not they were exposed to something or whether they were not exposed to something. And my immediate reaction is always like super positive, like, yeah, let's do it. But then I got thinking, like, Daniel, don't do it. If you write this function for this very bespoke case, then you have to maintain this function for the rest of your life. All right. And who wants to do that? So I said, okay. While I said yes initially, I got to thinking, how about I give you the tools to create this table very, very easily yourself. And then from there, you can make your own package that has TBL, Cochrane-Mantel-Hansel, CMH. So let me show you how to do that. So essentially what I told them to do is said, all right, so let's just break this table down into its very, very simple parts. Let's start with the orange here. This is like a TBL cross. All I'm doing is cross-tabulating two variables, exposed and not exposed, within the control group by their T stage. And then crude is the overall row. I'm just changing the label from overall to crude. So that's that one piece of the table. So what I can then do is use things like TBL strata to do that by cases and controls. So now I'm doing it two times. I'm just merging those two things together. So you can see that blue across the top, TBL merge. If I'm doing it for multiple variables, I can just iterate over those variables and do that TBL cross and that TBL merge independently, and then just stack the results. And at the end of the day, I can use something like add stat to just merge in some customized stats of those odds ratios, those Cochrane-Mantel-Hansel adjusted odds ratios, and the p-value. So this is a way that you can break a very complicated table like this into its simpler parts and move on with your life and just build a wrapper for new functions like this. So this is something that in every group I've ever worked in, we're always building our own bespoke versions of functions. So to build whatever we need to do. And this is just one example of how you can do that. So cobbling tables together. So most of the tables that we create in the medical space are pretty standard or standard adjacent. But it's often that you need like customized tables. And so those can be much more difficult to create for sure. So this is a quick example of how you can modify that underlying table body or that underlying data frame that comprises of what we're printing. So just look at this code right here. We're taking a data set. We're making a very, very simple summary table. And then what I'm doing here is I'm plucking out the table body or the data frame. So I hope you can see here or recognize that this label column is that characteristic labeled column in the printed table that you see. It has age, ethnicity, and it has the ethnicity levels right here. And then stat 1 and stat 2 are my placebo and active treatment arms, where you're getting the summary statistics for each one of those arms here. So this is just to drive home the fact that this is just a data frame. So don't be scared. You can always go in and modify this data frame directly. There's a function called modify table body that helps you modify this data frame in a way that still maintains the Gtsummary integrity of the table, because you could do some things to kind of destroy it. And then when I go to print it, I'm like, wait a minute, where did all the columns go? This using a function like modify table body will help you not do that. So again, this is just a summary table. It's just a data frame. Don't worry about it. You can modify it to get your really complex things to work for you. So I want to talk about ARD first tables.
Move on. There was a question like 2 sections ago I didn't get to ask, but it also relates to ARDs. So you had a slide up that was about comparing to our ARDs to confirm results, and said that you were working on functionality for cards and that. But the question was like, why do we need something special? Does running identical ARD1, ARD2 not work as expected?
Not entirely. So if you do run identical, you get a true and false back, right? And sometimes what you want from a comparison is not, was there any difference, yes or no? You also want to know what the differences were and how they were different. So that's one thing to consider in that space. But also, the formatting functions that you apply to your raw statistics. So like, for example, when we had our proportion that was 0.012 as the raw rate, or the raw percent, excuse me, the raw proportion, and then it gets formatted by scaling it to 100 and presented in the table as 1.2. Every time you make a function, I don't know if you know this, it was new to me at some point as well, but the environment is attached to that. So what can happen is that you can have 2 identical results of the raw proportion and the formatted one, but the formatting function that you're applying to it will have a different, it won't be identical because it has a different environment attached to it. So that's just one way that it won't work out for you as well, which is kind of like quite technical in some ways, but there's a lot of things that are hiding in the background that can make it a little bit more difficult. But primarily, I think when there are differences, you want to see what the differences are, and then using a function like that makes it very, very simple. But that's a good question. All right, let's talk about ARDs and building ARDs first. So a lot of our most basic functions in Gtsummary, TBL summary, TBL hierarchical, have a kissing cousin, depending on where you're from, TBL ARD summary or TBL ARD hierarchical. And these are data functions that do exactly what TBL summary does and exactly what TBL hierarchical does, except for rather than taking a data set as input, it takes an ARD as the input. Now we'll go through some various reasons why that's actually a really nice way to work. But before we do that, let's just go through a basic example to recreate something that's actually easier just to create with TBL summary. So this is, again, all from the cards package. We're not really talking a lot about cards today, but I just want to highlight some of the things that you can do with it. So in this example here, I'm using this ARD stack function where I'm giving it the ADSL data set. Let me just zoom in a little bit here. And I am doing a basic ARD summary for variable age. So that's a continuous summary. I'm doing a basic tabulation for ethnicity. And an ARD value is kind of like a dichotomous variable, which is going to give you the ARD for that single result, that single line result. And then these are a couple of options you can add just to make it a little bit cuter on the back end when you eventually do make your table. And you're going to get this rather large ARD. It's 38 rows and 9 columns, where you can see that you're getting for variable age, you're getting the n, the mean, the standard deviation, the median, the quartiles, the min, and the max. For a variable like ethnicity, at this Hispanic and Latino level, you're getting the n and the percent. And if I showed you more rows, which I didn't, you would see all of the levels for ethnicity there as well. So you can see that there are 28 more rows, and you can use print n equals ... to see more rows. So you can pre-calculate these things and build tables with them. So if I just took that ARD that I just built and passed that object to the TBL ARD summary function, I'm going to get a table that looks very similar to what we saw from TBL summary, but just giving it a data set instead. All right. So that's just an example of how you can use it. But why is this helpful? Oh, never mind. Let's do something more complex, and then we'll talk about why it's helpful. So here, I'm just essentially following the same structure where I am starting with my ADSL dataset. I am then labeling, giving my age variable a new label because that will get used later. I'm moving into ARD stack where I want to do things by arm. And I'm going to also change the default formatting function. So in this example, I'm going to have the standard deviation printed to 2 decimal places instead of the default, which I don't remember what it was. And then I'm going to go down here to my TBL ARD summary, and I'm going to say, okay, well, instead of that basic summary I got before, can I get a continuous 2 on 2 or more rows for my continuous variables? Can I get these statistics presented? And I don't want to see any missing data. I don't care. And then here I'm actually using modify caption to change my table's default caption. And you get this more complicated table. All right, so any ideas why creating ARDs to create first would be helpful? Any discussion? Anyone interested? Oh, and then Shannon is reminding me to take a break. Thank you, Shannon. This is the part I'm the worst at. I could talk forever, as you've seen during the exercise. Oh, is this a good start to? Maybe this is a good time to stop now. Let's take a 5-minute stretch break. Does that sound reasonable for everyone? All right. So you can just see here, you got your more complicated table, and you still did an ARD first. So there's still flexibility that you saw before. Blah blah blah. That's the end of the section. Wonderful. Thank you, everyone. I'll see you in 5 minutes. Can I just start a 5-minute timer here? All right. You're seeing the timer, right? Okay. If you have any questions in the pop them into the chat, and I can answer them during the break too. All right, let's return. Shannon, thank you so much for that. That was really lovely for me as well. Appreciate it.
I had a
Okay.
question.
Yeah.
A couple slides ago, I thought it was interesting that the ARD functions had formatting. Um, and then your table summary functions also have formatting, so you have formatting in two places.
Oh, good point. So if you are building ARD first, then the formatting is specified in the ARD. So there is no, in ARD, in TBL ARD summary, there's no digits argument to change the formatting, I believe. I may have added it at some point. I don't know, but it wasn't there originally. Um, so really what this helps us do is ensure that the ARD has all of the information that is going to then appear. So we want the ARD to have the raw value and the formatted value. So we take that information and go forward into the summary table functions. That's a good question. All right. ARDs are cute. Love them. All right. Let's talk about a little bit about table shells. So if you are building ARDs first, ARD first is a really nice way for creating table shells if you need to in your life. Um, it's not uncommon. So one way you can do it is, again, just to what Shannon was saying, is actually updating that formatting function in the ARD. So you can build your ARDs. And one thing I didn't tell you about ARDs is that they work even if you have no data, even if your data is garbage. So for example, if I am doing a tabulation on ethnicity and it's a factor and I have no data, what's going to happen is I get an ARD back that's always the exact same structure as what you expect to get back. And it's going to have 0 counts everywhere. And so if you tried to build that table, you would just get a table that had just 0 counts everywhere. So that's really nice because even when you have no data, what have you, you can still build ARDs. And there's a set of mock functions from the cards package that help you build ARDs from absolutely nothing, not even a blank data frame. So in this case, I'm just adding this one line. For my statistics for N and P, for my counts and my proportions, I want the formatting function to always return XX. Just no matter what the number is, just give me XX. And let's see what that ends up looking like. So then it ends up looking like a table shell. So this has been very common in the pharmaceutical space for a long time. Do you specify what you're building before you actually go through and build things? I don't really love this anymore because you are losing some context here by doing table shells. For example, what if my categorical variable, I needed it sorted from highest frequency to lowest frequency? If I just have a bunch of X's everywhere, that's not clear. So what I would recommend for table shells, rather than putting X's everywhere, is I would recommend um, just simulating some data. If you need a data set, or just using, if you're in the pharmaceutical space, you can use the pharma versus atom data sets, which are available and ready to use. They're really great. But with AI, it is just so easy to simulate data these days. So what you can do is if you just take a screen grab of this and said, Claude or Gemini or whomever your best LLM friend is, uh, can you simulate some data that would make this table? And you can specify what you want the column names to be. It will just simulate and it will look like this. So even if you gave it an XXX table, it can do the same. So what I like about that, again, is I said that there's some context that you lose from switching from actual numbers to X's. So like, for example, you can put things in ascending order, and that's clear by numbers, but not clear by X's. So I personally appreciate using actual simulated data or fake data to build my table shells rather than this method, but you do you. The print engines. Now that I've said print engine, it reminds me of something I was supposed to say earlier. Okay, I got it. It came back to me. All right. I will say it in the next slide. Print engines. So one of the things I think that has made Gtsummary very powerful and flexible and popular over the years is its ability to harness any one of these popular packages to print your table. So obviously GT is in the name of the package. That was the original printer. That's my favorite printer. It might be your favorite printer. And that's the one we started with. But you can also convert any Gtsummary table to a flex table, to a hux table, to a cable extra cable, or even just to a straight up table. Now there are benefits. Each one of these printing engines has its own strengths weaknesses. Like, for example, flex table, I think is the best table to print things to Word of all of the options. Do I think they're a little bit uglier by default? Yeah, that's okay though, because it has a lot more flexibility than, for example, GT going to Word. Hux table I think is great for PDF and LaTeX, and same for Cable Extra. I think Shannon, one of your favorites is Cable Extra straight to PDF, right? So that's a really nice options you hear. And you can see that each one of these output print engines has this function as GT, as flex table, as Hux table, as Cable Extra. And what that will do is will convert whatever Gtsummary you have into that object. So like I said, Gtsummary tables are what, two things, a data frame and a list of how to print it cute. So what it does is it takes that list, and if you're printing to flex table, it's going to convert that list of cute print instructions to actual flex table code and apply it to your data frame and give you a flex table at the end of the day. I'll show you why that's important. So in Gtsummary, we are not trying to give you every single way to customize your table, because these very complex and very, very diverse print engines already exist with tons of functionality. So let's just imagine that you have very, very specific output requirements for the width and the alignment of your table that you're outputting. You can convert your table to GT with as GT, that's line 4, and then you can utilize GT functions calls width and calls align to then specify exactly which columns are going to be which width and which columns are going to be aligned in various ways. Now, since we're writing this slide originally quite some time ago, we do have alignment in Gtsummary as well, but bear with me. Now, someone did ask the question, can I change this marking from a 1 to some other symbol? There is a Gtsummary function, a GT function that controls the marking symbols. And so you would convert your table to GT, then you would go and look up what that function's name is, and then you would use it. And then it would change those markings from the default 1, 2, 3, 4, 5 to whatever they are in whatever you've chosen, symbols, numbers, letters, what have you. Okay, so we've talked a lot about Gtsummary today. A lot, I would say. I want to go over some ways you can really adopt Gtsummaries for your group, your organization, your company, what have you, and what can ease that adoption process. So I think one of the best ways you can do that is with a company theme or a group theme. So every group that I've worked in that has used Gtsummary, we've had our own theme. That even applies to when Gtsummary was first, published and had all its defaults. I didn't think that what my group was doing as some of our defaults really should have been the default for everyone in the who was using the package. So I kept the defaults more general, and then I even had a theme for my own group, right? So I wrote almost all of this package, right, and specified what the defaults were, and even then I'm using a theme to change some of the default behavior. So themes control default settings for existing functions. They can actually even give you more fine-grained customization over what's not even available in arguments. So we have some themes that ship with the package, but you can also create your own. So just a quick example, this is the default output for a GT table. So sear this into your mind. We're going to show you some ways that we are going to change this table by simply setting a theme. So in this case, we are setting the theme Journal JAMA. So there's a few journal themes in here that will align your results with the reporting requirements of that specific journal. So like there's New England Journal, there's JAMA, Lancet, Economics Quarterly, I think it was called, some other ones. And this is the exact same code that you saw previously, except for the title's been changed. So what is different here? You can see here that instead of having a footnote that says the median IQR and N in percent, that has been moved up into the variable labels. So you can have your variable label, comma, and then what statistic you're seeing there. So that's just how they do things, right? The same for N in percent. So that got moved up. There's an N dash here separating the quartiles instead of a comma. And you can see here that the percent no longer has a percent sign because JAMA doesn't want to see any percents anywhere. All right. So This also applies to regression summaries and a few other things, but this is our basic table, and that's how, by just setting a theme one time at the top of your script, you can get tables that adhere to the journal's reporting requirements. There are language themes. So there are a number of languages that are supported. There's a couple of more that I'm supposed to review in pull requests at the moment, but it's hard to review language themes because I don't speak the languages. So, and I also want to do some due diligence that someone's not going to change like the word p-value to something naughty, you know. So it just takes a long time to review those pull requests. Although it would be kind of funny if I did that, if I just accepted it without checking. But anyway, this is an example of changing to Chinese. So there's a bunch of languages here. So I think this is really nice for, you know, presenting data in your own language, wherever you're at. There's one of my favorites is the compact theme. And what that does is it reduces the font size and reduces the padding. So essentially, every table I've ever created, I've reduced the font size and the padding. Otherwise, it just takes up so much space by default using the default settings in these print engines. So this is what I pretty much set every time. This is a great one. So you can see how cute and small this cute table is. All right, so now we're going to shift gears a little bit into a package that is available on CRAN, but is essentially for reporting at Roche and Genentech. All right. So I want to just show you an example of this package called Crane. And I want to just highlight some of the ways that we've built on top of Gtsummary for our specific reporting requirements. So the first way we did is we exported a new function called TBL Roche Summary, which is a very, very, very thin wrapper of TBL Summary. So what does it do differently? All of our continuous variables are going to default to continuous 2. TBL Summary reports missing values, but at Roche, we always report non-missing values. So all of the missing arguments have been renamed to non-missing, and they have different defaults, of course, so that report non-missing things. And instead of reporting 0 counts, but with 0 and 0%, we just report a 0. Okay, so some very basic things, but these are the kinds of things you don't want to be doing for every single table, because this is something that would apply to every single TBL summary table created at Roche. And so we just wrote our own. It's a very thin wrapper. You write it once and you can just reuse it again and again and again. So here's a Roche summary table. Oh, let me go over the code a little bit more. Here I'm adding a factor level that is not present in the data set. And you can see here that this non-missing argument is previously called missing in TBL summary, but in Roche summary, it's called non-missing. It says, always please report to me the number of non-missing observations. You can see here that now age, these are the non-missing observations reported for age. Oh, and we report them at the top rather than at the bottom. And you can see here that it has a default mean standard deviation, median, and min and max versus just median IQR. So a different set of default statistics. And for ethnicity, you can see here that now we have this refused unobserved level of ethnicity. And instead of 0 and 0%, it's just reporting it as 0. So a couple little itty bitty things, not a big deal, but would be kind of cumbersome to do every single time, if you ask me. So wrapping functions is a very good way. So if you are going to be using this within your group, I highly, highly, highly recommend that you create a little package as a companion to Gtsummary that does these types of things for you. So another common thing that's reported at Roche are our lab values. So if you ever enroll in a trial, they will take your blood and they will measure all sorts of things, right? So here's an example where we're just going to be showing albumin levels, right? So rather than doing a TBL summary showing me what my albumin levels were, the summaries at each of the visits on the trial, and what the change from baseline summaries were, which is 2 very easy things to calculate with TBL summary, and then putting them together with the TBL merge. We just wrote one function called baseline change, and what it does is it just puts it all together for you. So this is what the result looks like for, again, albumin results and using our lab data set. And this is the table. It's not that exciting. This is the value at the visit right here. And you can see here at baseline, there were 86 observations. The mean albumin level was 39.84. I actually think, yeah, I can't remember if anyway, these actually might be a different lab than not albumin, but anyway. And you can see here that the blank row here for change from baseline, because this is the baseline visit, then at week 12, it's repeated, but again, showing change from baseline. So all this is, again, is a TBL summary for the analysis value. That's what they call the value of the lab in academic data sets in the pharma space. Then doing another TBL summary for the change from baseline variable, and then just merging it with TBL merge. So that's all this is, but it just happens so often that we just wrote a function for it. So when you are doing this for your group, identify functions that you can very thinly wrap that will just make your life easier. New functions that you do so often that just having that function available to you will be super helpful. And then there's a couple, and then we're going to talk a little bit more about company theme and what we did at Roche. So we also in Crane created a theme called Theme Gtsummary Roche, and it does a couple of different things. It sets a custom function for rounding percentages. So at Roche, everything's presented to more decimal places than I think you probably need, including the percentages. So those are always presented to 1 or 2 decimal places. I can't remember what the default is right now. All p-values are rounded to 4 decimal places. This one hurts my soul a little bit because that is just not needed. 4 decimal places for 0.9998. Like those decimals at the end mean nothing, but whatever, they're there. That's what we do. In headers, in TBL summary, there's bold syntax, but in Roche, we don't want any. So we have no bold in any of our headers. So that's what you see here. All tables are printed with flex table because we are going to Word. And so, and we do update the font size, the table, the borders, the cell padding to meet all of our internal reporting requirements. So let's set the theme for Roche and run that exact same table that we've seen before. And this is what you get now. This is printed with flex table. Now you might be thinking that is a much uglier version of a GT table. And you're right, because this is a flex table printed in HTML. And the reason why we're using flex table is because it looks really nice in Word. So that's why we're doing it in Word. I love flex table, but GT is my favorite for HTML. Flex table is my favorite for Word. Different strengths, different spaces. So this is what you do. So if you are going to be using this at scale, I recommend you wrap functions with your own defaults. If you find yourself constantly making cumbersome changes to the defaults of Gtsummary, I recommend you build new functions for things that you do quite often. And I highly recommend that you create a theme for your group or your company. Also, you'll notice that what we've presented here is almost all univariate statistics and counts and proportions. Now, what about doing real research in a real research setting? Yeah, okay, that's good for getting table 1 and some really simple tabulations. But when you're doing efficacy tables, these are often based off of very complex statistical methods that are going far beyond our standard rates, counts, and univariate descriptions of statistics. So there's a whole vignette on how to create ARD first functions. So I recommend you read it. But just to give you a quick example, and we're not going to go over the details here, in Crane, we also export a function called TBL surfit times that would give you, you can specify multiple times, but we're just going to do 1 at 12 months here, that we want by treatment some of your basic time to event information. So you're passing a data set here, and there's some other defaults here that I'm not going to talk much about, but essentially it's building a surfit object in the background. It's building an ARD of that surfit object. And that ARD is going to have things like the number of subjects who are at risk at 12 months, what the survival rate or event-free rate is at 12 months, and the confidence interval for that rate as well. And there's other things there too. Now, looking at this, does this structure look familiar? We have a header, and we have here indented rows with things that are kind of related to one another, right? That looks very, very much like the structure of a continuous 2 summary from TBL summary. So what you can do is you can take that basic ARD of a much more complex model like a surfeit object in R for survival results. And because you can just take a basic ARD and pass it to TBL ARD summary and make these same things, what you're doing here is you're doing the exact same thing. You have an ARD of your results that are much more complex, and you're saying, oh, can you give this to me in the continuous 2 format for these results? And it will just give you the results in this format, and you specify which stats you want to appear. So that's how you can really utilize this ARD first framework to do much more complex things that are not available to you in like the most basic Gtsummary tables. As like the last resort, if what you're doing doesn't really fit into any of those frameworks that we've been talking about, you can just calculate statistics any way you want with an ARD, without an ARD, it doesn't matter. And you can just make a data frame. And there's a function called as_Gtsummary. And you can convert any data frame into a Gtsummary table. And then you can take advantage of all of those print engines and all those modify functions to add more, to style it, what have you. And then you know that at the end of the day, when you print all of your functions, that it's all going to have very, very similar styling because they all came from that same Gtsummary origin. All right. I hope that was helpful. Really, if you are using this at scale at your organization, you really should be familiar with these various ways of adapting Gtsummary for your own needs. All right. I want to touch a little bit about AI because, it's 2026. We've got to, right? So if you are going to the Gtsummary website these days, you'll maybe have noticed a cute little robot in the corner that said, and if you click on this cute robot, it's going to open up a dialog box where you can just ask. It's a chatbot. It's been specially trained on the Gtsummary documentation and supplementary documentation as well. So definitely go check out that chatbot. When you have questions about how to use Gtsummary, just go ask the chatbot. It's really helpful. The one thing that it does not do well is that it's quite constrained. And like if you start asking it questions where the answer might be do some pre-processing on your data set, then do the Gtsummary thing, it may not know that because it's really focused heavily on Gtsummary documentation, which doesn't always include things like pre-processing steps. So really, if I were a better maintainer, if I were a maintainer with maybe more free time, I can go to kappa.ai, which is running this chatbot, and I can see the questions that are being asked that are not being answered well. And I could go add supplemental documentation to address those questions, and then Kappa can learn from them and still give better answers. It's just hard to find the time to do everything. But this is still a very fantastic resource for just asking questions. There's a lot of documentation on Gtsummary, and now that these chatbots are available, I wouldn't read any of it. I would just go straight to the chatbot. But also, if you're going to file an issue or ask questions, you better have read the documentation first, just to be clear. So how else can LLMs help us with cards and the Gtsummary framework? So I didn't show the graph here, but so Gtsummary is actually the most widely used summary table package in the entire R ecosystem, which means there's a lot of Gtsummary code available on the interwebs out there. And so even things like ChatGPT and Gemini with no additional training are really good at answering Gtsummary questions out of the box, no special instructions. So here's an example. Can you act as a statistical programmer, and can you provide me the code for creating a summary table using ADSL? And I want to do age and age group, and these are the stats I want to see in that table, and just ask it to do it for you. So, and it will just give you all of the results, and it will do it in a way that's much more commented than any code I've ever written as well. So, it's actually simulating some data that looks like ADSL, like according to like the pharma standards, which is great, easy to do. And then it walks you through how to create that table. And again, it's giving you all of the options that are giving you exactly what you need, what you specified, and what are common in the pharmaceutical space, because it also has that context as well. So here you can see it's just giving you code to do it all. It's great. And then even at the very end, it's like, oh, if you're going to Word, I might recommend using Flex Table to print. But if you're doing HTML, I recommend GT, which is exactly what I would recommend. And I've never written it down anywhere, but it's somehow new. So they're really powerful. Highly recommend you utilize these tools. Why are ARDs important in this workflow? Is because ARDs, while we showed examples of them being R data frames, they can have language agnostic formats like JSON and YAML as well. And LLMs are fantastic at ingesting this type of data, JSON objects, and being able to understand it very well. So let's do an example where we take a quick summary table, we pluck out our ARDs, and we apply our formatting functions, and then we convert it to a JSON object. And so what that's actually going to look like is this very, 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 very long object that is, of course, going to be like, oh, for variable age group, we have variable level greater than 64, and I have a statistic called stat, little n for that, and its value is 221. This is a numeric value. I also have a formatted version of it, which is 221 character. There were no errors or warnings when I was creating that variable. I have the big N with the exact same thing. I have my proportion here, which the raw variable is 0.8701. My formatted version is just 8.8. It's just 87 as a character, and on and on and on. All right. So that's what the JSON representation looks like. And then just ask your LM to write a summary. So below you'll find a JSON representations of a results table. Act as a medical writer in a pharmaceutical company and write me a brief summary of the results. And it gives you a lovely summary here. So very, very easily done. Very, very nice way to work with LLMs. I want to end with my new favorite way of doing this, and it's using like a skills MD file with Claude. If you, and I think a lot of the code engine AIs have something similar to skills MD, but this is a way to have like a coding parser that really adheres to what you want it to do. So, for example, if I am using Crane, I want Claude to use Crane if there's a function in Crane to do it, because I have standardized my things in Crane to do exactly the way I want it to do. So I want it to use Crane functions, otherwise use Gtsummary functions. I want you to extract the ARD immediately from every table you create using gather ARD. This is how I want you to create tables. If the data first way of creating tables doesn't work or it doesn't exist, then path B, do an ARD first method where you can calculate the ARD with cards or any kind of ARD function from cards, then use one of the Gtsummary TBL ARD functions to build a table. I always want you to use tidyverse for data prep. I always want you to load my theme at the top of this so that I get the same styling that I'm expecting. And you can just drop this in one place for your whole team, and your Claude coding assistant will just do what you want every single time. It's beautiful. I love it. So have a good time. All right. We're gonna wrap up. So before we start the wrap up, are there any questions? If you have more questions, you can still interrupt at any time or at the end. So check out there's supposed to be an iframe of the website here. It's not here. I don't know why. But check out the website. It has a lot of great documentation. It has the AI chatbot in it. It's a great resource. Just want to thank all of the contributors. You may also notice that Shannon Pileggi, thank you for being here, Shannon. She's an author. Emily Zaber, our wonderful moderator for today from Our Medicine. She's also an author, been there since day one of Gtsummary. So thank you, especially to you two for being here today and for your help with the package. And there's been so many ways that people have contributed to Gtsummary with pull requests, with ideas. It's been a huge community effort. So just a big thank you to the community. I should update this slide. Instead of asking on Stack Overflow, I should say ask your AI friends. But all right. But anyway, thank you for being here. Thank you for coming. It's always lovely to connect with people. So really appreciate that. And if you have questions, I'll stick around for a few more minutes to answer any questions you may have about using the package or extending the package for your own use cases.
That was amazing. Thank you so much.
Thanks.
I use the package almost every day and I learned a lot today still, so.
Oh, nice. Yeah, I think since we started adopting it for Roche, we did make a number of updates that were quite nice. All right. 2 hours of tabling was enough for everyone. That's good. All right, no questions. thank
A
you everyone
lot
for
of
coming.
a lot of thank yous, no questions.
All right. Oh,
Do you
Eva,
ever
if you
hand
go to
out hex
Posit,
stickers at conferences?
yeah, I'll be at PositConf this year.
But do you
So
print hex stickers?
I do. They're very cute.
Okay.
I sometimes forget to bring them to the conferences after printing them, though. I will say that.
I think everyone has a lot to think about now. A lot of nice new features over the past couple of years. Thank you so much, Daniel, for everything you've done.
Oh, you're very welcome. It's been a lot of fun. All right. Should we close it down?
Yeah, I think that if there are no questions, we can say goodbye and thank you again. Very, very useful. All right.
Thanks.